When talking about the different mechanisms of horizontal gene transfer, I like to start with transformation just because it's kind of the most straightforward. So all this is basically taking up DNA from their environment. So it's not from another cell, it's just out there floating around for some reason, and the organism can take it up. So this can be segments of DNA, or it can also be whole plasmids that have been released from a cell. So don't get this confused with conjugation. If it's not in a cell, it's still transformation, even if it's with a plasmid. So this naked DNA that's kind of floating around in the environment can come from like a lot of different sources. Usually it's associated with a lysed cell. So naked DNA is just DNA without a cell surrounding it. It can come from a virus lysing the cell. It can be released from the cell if they're like kicking out some genes they don't want for whatever reason. That's pretty rare. Usually it's from some sort of lysis, either virally induced, or we were talking previously about like osmotic imbalances and how that can lead to cell lysing. That's another way that this DNA can get out of the cells. And so another thing to note is that it's not that all bacteria are capable of just going around picking up free-floating DNA. You have to be what's called a competent cell. And so there have been a lot of studies that have kind of looked into this. Of course, they're a little bit biased towards taxa that we're familiar with, but we know that there's a large taxonomic range of species and groups that are capable of doing transformation. So the way you become competent as a cell is basically you have to be able to detect that there is DNA in your environment, which is pretty common because again, as I was saying in the previous video, this is advantageous to bring it in for several reasons. You then have to be able to bring it in through the cell envelope, which of course there are different mechanisms for gram-positive and gram-negative bacteria to accomplish this. But you have to bring it into the cytoplasm, and then you have to incorporate it into the genome. So if you don't have all of those abilities, you would not be considered a competent cell. The other thing, when we talk about biotechnology, a lot of times we'll talk about making a cell be competent. That basically means making it receptive to these things. So a cell could theoretically be competent, but if it doesn't need that foreign DNA, it's not just going to incorporate it. So you kind of have to stress it out sometimes in the lab to make it willing to accept this foreign DNA.